Good morning, everyone. Am I audible?
Yes, yes.
Okay, I'll take electrophoresis. Can you see the slide? Everyone can see the slides. So this is basically practical topic, but in exam you don't have to do this procedure. Okay, this is visible. Is it visible? Okay, we will discuss about electrophoresis. I think everyone of you must have heard, even if you don't know about it. So before starting electrophoresis, these terms you should know, and I think you will already know. It's a little confusing, but I think you know, because when we will discuss electrophoresis, you will see these things. Cathode is basically negative electrode, but cation is positive ion, right? And zeta ion, you know that basically when this in a solution molecule which can have negative and positive both charges at particular pH, they will have net charge 0. So at that time, that is known as zeta ion. So the charge at which the net charge becomes 0, basically the pH is known as isoelectric pH, right? So what is electrophoresis basically? It is basically the migration of charged particle under the influence of an electric field. So basically if you are like passing electric current through some molecule which you have kept like in a medium, and then under the influence of electrical field, those particles based on its electrical charge mainly, it will start migrating. So different molecule which will have different charges and other factors also, we will see what are the other factors which influences this movement. We will start moving at different paces, and that way we can separate, we will be able to separate different molecules. So what are the important biological molecules which is separated with the help of electrophoresis, like amino acids, peptides, proteins, nucleotides, nucleic acid? You know that all these molecules have charge, right? And they possess ionizable groups. So because of that, these molecules can be separated by the process of electrophoresis. So these molecules can either exist as positive cation or anion depending on the pH. Now when in an electrophoretic system, when isoelectric pH is given, then the molecule will become zeta ion, net charge will be 0, but these zeta ion will acquire positive charge in a solution if the medium of the solution of the electrophoretic system is made acidic. So in acidic solution, zeta ion acquires positive charge, and in basic solution, zeta ion possesses negative charge. So what is the principle behind the process of the electrophoresis? It is basically movement of the solute or molecule is equal to electrical charge, basically electrical field which you are giving, into net charge, the charge of the molecule, here you see, divided by frictional coefficient. So basically this friction coefficient will oppose the movement of the molecule, charged molecule, but the electrical field which you are giving will promote the movement of the molecule, basically the charged molecule. And friction coefficient will generally mainly depend on the size, shape, size, and the viscosity of the medium. So if the medium in which you are keeping the solution solid for the movement, if it is more viscous, the movement will be slower. All these things are written in your manual also, you have to go back and read also. Now what factors this migration of this charged molecule in the process of electrophoresis will depend? It will depend first on molecular weight. So I think it is like you will understand easily that a small molecular weight particle will migrate rapidly, right? Then size and shape of the molecule, then electrical field strength. The more the electrical field you give, more the movement will be there. Temperature of the operation, the net electrical charge of the molecule. The highly charged molecule will of course move more rapidly. So now how, where, on what factors this, especially this charge, will depend? It will be affected by pH. I told that zeta ions are used, and based on the pH of the medium or the buffer used in this process, this charge will be affected. And another thing is ionic strength of the buffer. If buffer is such that the molecule becomes more ionized, then the electrical charge will increase. So this electrical charge will depend on the pH of the medium and ionic strength of the buffer. So molecular weight, shape and size, electrical field strength, temperature, then electrical charge, which depends on pH and ionic strength. So this topic will not come in, like you don't have to do this in a practical, but like there can be some like you have observed in another session those types now. So in those tables generally it will come. So these questions can come with that. So for this process, generally the apparatus will look, this is a symmetric diagram. So you see here, so this is a supporting medium on which you will put the sample basically, and that sample will move when you start give the power supply. This sample will move. So suppose like how this sample will move, because if you are giving this power, electrical current has to reach to the sample, then only the movement of this charged particle, whatever sample you are putting, will start happening, right? For that we keep here buffer. So these buffer chambers help in the transmission of this electrical current to the sample. So how this buffer is connected? Buffer is connected to the sample with the help of wick, basically filter paper. So this filter paper will absorb the buffer, and that will, along with the buffer, it will carry the electrical charge also. So once you start the power supply, this circuit will be completed. Charge will reach the molecule of interest, and it will start moving on this supporting medium, and depending on its various factors of migration, as we saw, all the molecules of the sample will move at different paces, and then it will separate, and we can study that. So so this is basically the electrophoresis unit. It consists mainly of supporting media on which you will put the sample, then buffer system to carry the applied current, and electrodes which will be connected to the buffer. And so supporting media can be gel electrophoresis, or it can be capillary. So the supporting media can be gel on which you can put the sample. So it can be slab gel, this gel, 2D gel, or capillary. Instead of the gel, you can we can take the capillary, like narrow bore capillary, and through the capillary also sample can be carried. Buffer system, it carries the applied current, and it determines the charge carried by the solute. So basically buffer will be carrying the current, and it will determine the charge, as I told that the charge will depend on the pH and ionic strength of the buffer. So now this method, that was the electrophoresis unit and method of electrophoresis. So it can be either manual, semi-automated, or automated. So in manual and semi-automated, mainly gel is used. So two important gel are agarose and polyacrylamide. Even cellulose gel is used. So these are gel, and in automated, capillary. Mainly capillary is used in automated electrophoresis system. So just know the name. If someone asks what are the different methods of electrophoresis, can manual, semi-automated, automated. So see this again, and you will be able to understand. This is the power source. Here you see electrodes are there. It is connected to the buffer is connected to this supporting media, which is gel. With the help of wig, filter paper, this will, once you switch on the power, it will carry the current. This buffer will carry the current to the supporting media, which is carrying the sample. Here sample value you can see. With the help of this wicks, filter paper, once the current reaches the sample will start migrating. So thus migration will depend on all the factors we read, and then it will separate, and then we can read by staining the. Now types of electrophoresis depending on the direction of run. So just now we saw this is horizontal, right? So it can be horizontal or vertical. Most of the electrophoresis, commonly conventional electrophoresis, are horizontal, but some can be vertical also. So horizontal electrophoresis, what is the supporting media, most common supporting media? So you have to know for serum protein electrophoresis. In exam questions in practical, they will generally ask related to protein electrophoresis. So the protein, most common type of protein electrophoresis is basically agarose gel electrophoresis. It means agarose is used as a supporting media. Agarose is linear polysaccharide made of agarobioes, galactose and anhydrogalactose. This is a repeating unit of this agarobioes, and this is the most common supporting media used for protein electrophoresis. What are the reagents required? We need one buffer. We saw that what is the use of utility of buffer. So tris glycine buffer is used, and pH is 8.6. Why 8.6? At this pH, at this alkaline pH, all the protein molecules, because we are taking suppose serum, so all different protein will be there, albumin, different types of globulin. Okay, so those molecules acquire negative charge. So because of that, all the molecule have negative charge, and depending on the size of it, it will start separating. Then agarose gel will be required. Then next, acid cold ethanol is required. This is for fixing. Once you have run the sample after giving a power supply, electric current, suppose for certain amount of time, and after that you have to fix that sample on the slide, then only you will be able to read it. After fixing, you have to remove the extra agarose and other things, so you have to use acetone for dehydrating. Then we will use the staining. So whatever sample is left on the slide, we will stain with the amido black dye, and then during the run itself we put mix the sample with the tracking dye, bromophenol blue, because once we start the run, sample will start moving from here. We will not know, because it will keep moving if you keep giving electrical current, electrical supply, and then the sample will go out of this slide itself. So we have to know that it has reached, okay, suppose near the opposite electrode, and then we have to stop the run. Otherwise all the samples will fall only. So that's why you have to keep tracking dye, that is the colored dye. So that dye helps in tracking the movement of the sample. Supporting media, I told agarose is most common, but other supporting media used are polyacrylamide, then cellulose and starch. Procedure. So first you will layer 4 ml. This is the typical conventional manual method of protein electrophoresis, agarose gel protein electrophoresis, which we normally do in lab. Okay, so if we were here we could have shown you, but right now it's not possible. So you have to layer 4 ml of agarose gel on a slide. You have to take glass slide, and this agarose gel, which is going to be your supporting media, you are going to put that on the slide evenly, and then it will take 3 to 5 minutes for setting down. Then at a separate place you have to mix the sample with the tracking dye. I told you what is the utility of tracking dye. And then that tracking dye and sample is basically we have to cut a filter paper, Batman filter paper number 42. Even if you don't remember, that is fine. Small spikes is cut, and these sample and tracking dye is soaked in that, and then that filter paper spikes is kept on the supporting media. You saw how we kept the sample, multiple samples. Okay, now this slide which is containing the supporting media and the spikes which contains the sample will be put in the electrophoretic chamber. Now both sides you will fill the buffer, and then with the help of Watson filter paper 1, that will act as a wick, and that you will connect the supporting media with the buffer chamber, and then electrical current will be applied, generally 120 volt, and it is run for 1 hour. After the electrophoretic run is complete, the slide is taken out from the electrophoretic chamber and put in acid ethanol for fixing the samples for 30 minutes. Then slide is taken out from there, and it is put in 90% acetone for dehydration. So it removes everything other than the proteins of, if you are using blood or serum. So whatever proteins is present in blood or serum, apart from that everything will be dehydrated and removed from there. And then the slide is dried in an oven at 100 degree, and once the slide is dry you stain with the amido black, and then it is washed with water or dilute acetic acid. So whatever protein is left, that will be shown only. So like this mostly you will see. So of course this is the color representation. Slide will be mostly white, and this will be generally blue color. The dye will stain the proteins blue. So what is happening, this is the origin, right? I told you that all the protein acquired negative charge at pH 8.6. so buffer UV used was 8.6. so then if all the proteins have acquired negative charge, it will start moving towards positive, right? Positive electrode. So the movement will start like this. Now albumin is the most negatively charged molecule and smallest also, so it will move the furthest from the origin. So this way all the proteins, the major classes of protein in the serum is separating: albumin, then alpha 1 globulins, alpha 2 globulins, beta 1 globulin, beta 2 globulin, and gamma globulin generally does not acquire charge, so it will stay near the point of origin only. So you have to know what are the proteins present in all these different bands of serum protein electrophoresis. In exam, like in observed and non-observed session, we can give this, and you have to write what are the protein present in these bands, like that, and when it will increase, when it will decrease, we'll see. So we'll see here what are the proteins which is present. So these are the protein: albumin, right? Then in alpha 1 antitrypsin is important. This is important. Then in alpha 2 mainly you will find ceruloplasmin, haptoglobulin, and alpha lipoprotein, even alpha 2 macroglobulin. So you have to know, remember the name, and I think in your manual everything is given. Then in beta region, the beta 1 and beta 2 both combined, generally you will find hemopexin, transferrin, and beta lipoprotein. So remember these things: hemopexin, transferrin, and beta lipoprotein. why we want to separate this, so that we can see, because we already know normally in how much quantity these different fractions of proteins are present. So if some band is decreased, like lighter or darker, depending on that we can understand that some protein is increased or decreased in the body. Then in gamma region, mainly immunoglobulin you will find, but in some conditions C-reactive protein you will find if infection, inflammation is present, and fibrinogen should not be there. I will tell you the reason why. So how you will read this? You saw this, but how will you understand how much is present? So the instrument used is densitometer. So the process is densitometry. This densitometer, basically if we put this slide in the densitometer, it will convert, basically with the help of, by the observance method, it will convert these fractions and it will form a electropherogram. We will see how it will look, and the machine will then change the will calculate the area under the curve of each electropherogram peaks. So it will look like this. This is the normal. Don't look at this for the time being. Now you saw that bands were there, right? So albumin band was there, alpha 1, alpha 2, beta 1, beta 2, and gamma. So those bands, by the help of absorbance, the densitometer has converted, depending on the intensity, it has converted those bands into the spikes. So like this, it is giving a spike. The albumin band was the most dense, so this spike is the tallest spike, and then it will calculate the area under this curve for each partitions of the protein, and it will give you the value, and we already know the normal value, and we can see that whatever percentage it is giving, if it is more or less, depending on that we can see that it is normal or not. So this is albumin, this is alpha 1, alpha 2, beta 1, beta 2, and gamma. So this reference range, it's okay if you don't know. Such question will not come. Normally we will give the normal pattern, abnormal pattern. You will have to tell which protein might be present in that abnormal pattern. So this is generally normal pattern like this. Albumin will be the tallest peak, and then other peaks will be like this generally. Now here you see that there is some peak in gamma region. So, and this peak looks very similar to albumin. So this is basically M protein. This is found in multiple myeloma. So this you have to go back and read in your manual. It is there, subsets of proteins present in different bands. This question is asked in practical. Now clinical interpretation, the most important is multiple myeloma. Multiple myeloma, then Waldenstrom, macroglobulinemia, and amyloidosis. In gamma region, there will be sharp peak that will signify multiple myeloma. Mostly in exam question come from multiple myeloma only. And then when gamma globulin is decreased or it is absent, then it means it is agammaglobulinemia, hypogammaglobulinemia. Means gamma globulins proteins body is not able to produce. Now albumin generally is found to be increased in dehydration and decreased in nephrotic syndrome, chronic infection, malnutrition, and impaired liver function, because albumin is produced from liver, right? So in nephrotic syndrome, why? Because albumin is a small protein, and as soon as the nephron damage starts, this will, the charge of the membrane of the nephron gets lost, and this protein being the smallest, they start moving out, getting excreted first. So albumin decreases in nephrotic syndrome. Alpha 1 globulins are found to be increased in pregnancy, and it will be decreased in alpha 1 antitrypsin deficiency. Alpha 2 globulin in nephrotic syndrome it will decrease, and in Wilson's disease, protein losing enteropathy and malnutrition, this will increase. So you have to go back and read this. Now you can you will be able to understand one or two conditions at least with the help of the proteins present. Beta globulin, iron deficiency anemia, definitely it will be increased, and then protein malnutrition, beta globulin will decrease. What is the limitation of electrophoresis? In this, plasma cannot be used. We have to use serum. Why plasma cannot be used? Because plasma contains fibrinogen, and it gives a band in the gamma region that will be confusing for the diagnosis, because the sharp band in gamma region is found in multiple myeloma. But if fibrinogen also gives band in the gamma region, then the diagnosis will become difficult. So that's why plasma is not used. For same reason, hemolyzed sample also is not used, because hemolyzed sample will also have free hemoglobin, and that hemoglobin also will give band in the gamma region. And lipemic sample we cannot see the separation properly, and the separation does not happen properly in the supporting media if example is lipemic. So these are the limitation of this process, gel electrophoresis. Abnormal pattern, some of the abnormal pattern you see because in exam it will come. So here you see what is happening. This is the normal and this is the abnormal pattern. Here protein loss is happening, selective protein loss. Albumin is decreased, right? And then alpha 2 is increased. Why alpha 2 is increased? Alpha 2 contains bigger molecule, alpha 2 microglobulin. So those molecule does not gets excreted in nephrotic syndrome. In nephrotic syndrome, selectively smaller molecules gets excreted. So albumin is decreased and alpha 2 is increased. Then your diagnosis will be nephrotic syndrome. In these slides I will give you later. Then here you see that most of the molecules are like different subsets of the protein are decreased. Okay, and there is gamma and beta bridging. So this is very typical of chronic liver disease or viral hepatitis or chronic alcohol abuse. So one of the diagnoses if you write, but the most important diagnosis which you have to write, if you see that beta gamma bridging is there, separation is not proper and albumin and most of the bands are reduced, then it is cirrhotic liver disease or chronic liver disease. Now if gamma globulin and alpha 2 molecule, alpha 2 globulin is increased, then it is chronic inflammation. And then chronic inflammation, of course, that happens during malignancy also. So chronic inflammation, chronic liver disease, malignancy, but most important will be chronic inflammation. When you find that gamma globulin is also increased along with alpha 2. So those molecules are increased because those molecules are generally increased to fight the infection and in the inflammation which is going on for a long time. So that's why in chronic inflammation these molecules will be increased. Hypogammaglobulinemia is very obvious. Then polyclonal gammopathy. Here the gamma band, the region in protein, you can see that it is more dense and albumin is reduced. Okay, so this is polyclonal gammopathy. Why it is polyclonal? It is not monoclonal. Monoclonal gammopathy is multiple myeloma. In monoclonal, the band will be even sharper. Here polyclonal because in the whole gamma region, it is diffused. In the gamma region is increased basically, like stained more in a diffuse way. So it means different types of immunoglobulins are increased. Right. That is the meaning of polyclonal. From more than one clone of the plasma cells, you will read in immunology that different immunoglobulins are produced from different clones of the plasma cell. So that's why different types of immunoglobulin is increased, so it is diffuse. So this is mainly polyclonal gammopathy present in hematological disorders, malignancy, connective tissue disease, liver disease. Why liver disease? Liver produces generally albumin and globulin is produced from plasma cells, B cells. So if liver disease, then relatively the concentration of the amount of the albumin will decrease and relatively gamma globulin will be increased. So this is multiple myeloma. This will be very typical. You will be able to understand easily because the band in the gamma region is similar to the albumin band. So this is basically multiple myeloma. And if it is given as electropherogram, we saw the how a spike looks similar to the albumin. So what are the applications? You have to know definitely for serum protein electrophoresis. Other lipoprotein like alpha, beta, pre-beta, these are the lipoproteins, right? You would have read in lipid chemistry and metabolism. Lipoprotein also you can separate. Then different types of hemoglobin we can separate, and we can see whether there is any hemoglobinopathy or not, right? With the help of pattern of the electrophoresis. Then in serum protein electrophoresis, alpha 1 antitrypsin deficiency, multiple myeloma, all these diagnoses we can do with the help of protein electrophoresis. Then genotyping of protein, not very important at your level. Then small molecules, drugs and steroids can also be separated. Then CSF analysis can be done, and urine analysis in glomerulonephritis and all. So what you definitely have to write an answer in application if it comes. So that separation of protein, isoenzymes and DNA fragments. What are the example of isoenzymes? Like CK or LDH, all this, right? So those have many isoenzymes, so those can be separated. Then hemoglobin separation, and third is lipoprotein separation. What are the other types? You just have to know one or two sentences regarding that. What we saw was agarose gel electrophoresis for serum protein, right? So other types are isoelectric focusing electrophoresis. Here the separation will be based on isoelectric pH, not on the charge or size and shape. Capillary electrophoresis, the basically supporting media is a small bore capillary tube. So this is useful for separation of low molecular weight protein. Very low molecular weight protein does not move. If all the like different protein fractions are of low molecular weight, the separation will not be proper in the gel electrophoresis. So capillary tube is used. And what is the advantage, that here sample required is very less. Okay, and then as I already told, that capillary electrophoresis is fully automated. It's easier to do also, and high voltage can be applied and separation can be better. So here also the like the basic setup is similar, but here you see that this two electrophoretic chamber is not connected to the sample. Sample will be kept somewhere here. Suppose in the tube you will inject the sample. It is not on the supporting media. It is a tube, capillary tube. Immunoelectrophoresis, when the sample you are using are basically antigens. So the separation of antigen, and you will give the antisera in the sample also. So that will be immunoelectrophoresis. With the help of antibodies, this antigen can be separated. Now for vertical electrophoresis, the example is SDS page, sodium didecyl sulfate polyacrylamide gel electrophoresis. So what it does, that SDH will give uniform negative charge. So basically SDS will make all the proteins negative. Once all the protein now is containing uniform negative charge, then the effect of the charge will not be there during the separation. So then the protein will separate only on the basis of size. So this is basically used to estimate the protein size and quantify protein. So vertical electrophoresis will look like this. Sample you will keep here, and depending on, because now all the proteins have equal negative charge, so it will separate based on the size. Now for hemoglobin separation, generally cellulose acetate supporting media is used. For protein it is agarose gel is used. For hemoglobin it is used is cellulose acetate. Here also pH is around 8.4 to 8.6 only, and this generally this technique can like with satisfaction detect different types of hemoglobin variants. So how the pattern will look, we saw the pattern mainly for protein electrophoresis. See the pattern for hemoglobin. So this hemoglobin A, the adult hemoglobin, right, is F, S is important, A2, then E, C. So like this it will separate, and depending on the band present in the sample, you can determine whether there is any abnormal or unusual hemoglobin variant present in the body or not. And just so you know, and you will read these things in patho and other subjects also, I'm sure, that what are the different variants. HbA is normal individual, I think everyone knows HbA, and F in neonates F is present, right, and increased F is present in beta thalassemia also, when body is not able to make like normal amount of hemoglobin A, normal variant, then body produces hemoglobin F. Beta thalassemia, I think you would have read, or you will read in theory also in biochemistry. Hemoglobin HbS, so homozygous HbS individual. HbA and S, it means it is heterozygous sickle cell. HbS&C, sickle cell individual disease. So for a like your practical exam purpose, I'm telling you, mainly you will come as charts. So there will be given some pattern, and some band will be marked. You will have to write what are the proteins present there, or some abnormal pattern we will give, and you will write what may be the condition. So for something you have to write exact answer, like if there is sharp band or sharp spike in the gamma region, you will have to write multiple myeloma. But if suppose there is increase in gamma region we are showing, and so we will give the normal and abnormal pattern both. We'll try to give that if it is something which is not very obvious, some disease. So you have to go back, read, everything is written in the manual. And normally for during one of the exam days, definitely one of the chart will come from this. And for practical like theory viva also, or for practical viva purpose also, you have to know what are the different proteins present in the different band, like alpha 1, alpha 2, when it is increased, decrease, those things you have to know. That's it for today. Okay, thank you. I'll stop the presentation.
Thank you, sir.
Thank you. Bye.